Take a moment to settle in. What follows is not a story. It is a record, partial, incomplete, and honest about its own incompleteness, of what exists at the very bottom of the world, and of every reason we cannot simply go there. There is a depth at which the ocean stops being familiar. It stops being the thing we sail across and fish in and draw maps of. Below 6,000 meters, the ocean becomes something else entirely. A zone of pressure so extreme that the physics of ordinary materials no longer apply, where darkness is not merely the absence of light, but a permanent geological condition, where the weight of the water column above would destroy any container not built specifically to resist it. This zone has a name. Scientists call it the Hadal Zone, derived from Hades, the Greek underworld. The name was chosen deliberately. It describes a place that, for most of human history, was as unreachable as mythology. The Hadal Zone occupies roughly 1 to 2% of the global ocean floor. That fraction is misleading in its smallness. 1 to 2% of 361 million square kilometers is still between 3 and 7 million square kilometers, an area comparable to the entire continent of Australia, hiding below 6,000 meters of water, largely unvisited, poorly sampled, and known only in outline. The detailed biological, geological, and chemical inventory of this zone is a document with most of its pages still blank. The zone is not uniformly distributed. It exists almost entirely within the deep ocean trenches, the places where one tectonic plate slides beneath another in a process called subduction. The Pacific Ocean contains the vast majority of these trenches, arranged in a rough arc around the basin's perimeter that geologists call the Ring of Fire. The Mariana Trench, the Philippine Trench, the Tonga-Kermadec Trench System, the Kuril-Kamchatka Trench, the Japan Trench. Each of these is a scar in the ocean floor kilometers deeper than the surrounding abyssal plain, created by the slow-motion collision of tectonic plates happening on a timescale of millions of years. The Atlantic has only one true Hadal Trench. The Indian Ocean has one. The Southern Ocean has one. The Pacific has most of the rest, which is why Pacific biology, Pacific geology, and Pacific ocean science have dominated the study of the Hadal Zone since the first measurements were made. To understand why the Hadal Zone remained unvisited for so long after its depth was first estimated, you need to understand something about the history of ocean measurement. The history is both older and more recent than most people assume. The first systematic attempt to measure the depth of the ocean across more than a few local patches was made aboard the HMS Challenger, which departed Portsmouth, England in December 1872 and returned in May 1876. The Challenger was a corvette converted for scientific use, stripped of most of its armament to make room for scientific equipment, rope storage, and the machinery needed to take measurements. The method of measurement was called sounding. A weighted line, rope, or wire, depending on depth, was paid out over the side of the ship until resistance stopped, indicating that the weight had reached the bottom. The length of line deployed gave the depth. A single sounding in deep water could take hours. The crew would pay out rope for an hour or two as the weight sank, then haul it back in, then move the ship and do it again. In shallow coastal waters, you could take soundings quickly. In the deep ocean, a single measurement consumed most of a working day. Over 3.5 years, the Challenger made 492 deep soundings, covering 127 through 600 kilometers of ocean. The data it collected was genuinely transformative. The Challenger expedition established that the deep ocean existed, that it was cold and dark and largely devoid of the organisms scientists had expected to find, and that its floor was not smooth but featured enormous mountains and valleys, comparable in scale to anything on land. One of the expedition's soundings, taken in the Western Pacific, measured a depth of roughly 8,000 meters, the first evidence of a deep ocean trench. The Challenger Deep in the Mariana Trench, which bears the ship's name, was measured more precisely by later expeditions. The point is that 492 measurements, however remarkable, represent 492 dots on a map that needs tens of millions of points to be called comprehensive. Echo sounding changed the mathematics of ocean measurement when it became available in the early 20th century. The principle is elegant. Send a pulse of sound downward from a ship. Measure how long it takes for the echo to return from the bottom, divide by twice the speed of sound in seawater, and you have the depth directly beneath the ship, because sound travels at approximately 1,500 meters per second in seawater. Even a measurement of 10,000 meters requires only about 13 seconds for the round trip. You could take thousands of soundings in a day rather than a handful. Ships equipped with echo sounders could map the depth of the ocean along their entire course, building up a picture of the seafloor's shape wherever they traveled. But the fundamental limitation remained. The sound gives you a single point directly beneath the ship, and covering the ocean with points requires the ship to be everywhere. When the first echo sounders were attached to Navy vessels crossing the Atlantic in the 1920s and 1930s, they revealed an astonishing feature: a continuous mountain range running down the center of the Atlantic basin, roughly following the shape of the ocean's coastlines. This was the first evidence of what would later be understood as the mid-ocean ridge system, the longest mountain range on Earth, running 60,000 kilometers through every major ocean basin. It was discovered by a ship going about its ordinary crossing, not by any systematic survey effort. Multibeam sonar, which arrived in operational form in the 1970s, transformed ocean mapping again. Instead of sending a single sound pulse straight down, multi-beam systems emit a fan of sound pulses simultaneously, covering a wide swath of seafloor with each ping. At a water depth of 4,000 meters, a modern multi-beam system can map a swath roughly 40 kilometers wide with each pass. This is enormously more efficient than single-beam sounding, but the arithmetic of coverage is still formidable. The total area of seafloor deeper than 200 meters is approximately 335 million square kilometers. To cover that with a 40-kilometer wide swath would require a ship to sail lines spaced 40 kilometers apart across every ocean basin, a campaign that, at reasonable ship speed, would take something on the order of several hundred years of continuous effort. This is a physical constraint, not a funding problem. There is simply a lot of ocean. The Seabed 2030 initiative, a collaboration between the General Bathymetric Chart of the Oceans and the Nippon Foundation, set an explicit goal of mapping the entire ocean floor at meaningful resolution by 2030. When the initiative launched in 2017, less than 15% of the seafloor met that standard. As of 2024, roughly 25% does. The remaining 75% is known only from satellite-derived gravity data, which provides a resolution of approximately 5 kilometers per pixel. At that resolution, a submarine, a seamount 3 kilometers wide, even a canyon system a kilometer across, all of these fall below the threshold of visibility. They exist in the data as faint anomalies or not at all. The surface of Mars has been mapped at better than 1 meter per pixel by the Mars Reconnaissance Orbiter. The surface of the Moon has been mapped at comparable resolution. Both of those worlds are hundreds of millions of kilometers away. The deep ocean floor averages less than 4 kilometers below the surface of the water we live on top of. The reason the ocean floor is less well mapped than the surface of Mars is not primarily a failure of technology or ambition. It is a consequence of physics. Radar, the tool that maps other planetary surfaces, cannot penetrate seawater. Electromagnetic radiation at radar wavelengths is absorbed by salt water within a few centimeters. Radio waves do not reach the seafloor. Satellites cannot see through the ocean. The only form of energy that penetrates seawater effectively over distances of kilometers is sound, and sound requires a ship directly above the area being mapped. There is no remote sensing shortcut for ocean floor mapping. You have to be there, physically, with acoustic instruments, moving slowly across the area you want to document. The Hadal Zone adds a further constraint on top of all of this. The deepest trenches are not merely inaccessible in the sense of being far away. They are inaccessible in the sense that ordinary sensors cannot reach them at all. A standard oceanographic instrument package, a conductivity, temperature, and depth probe lowered on a wire, can reach several thousand meters with relatively conventional equipment. But reaching 10 or 11,000 meters requires very long wires, very heavy winches, and very careful management of the enormous mechanical stresses involved. The wire itself, loaded with the weight of kilometers of cable, can break under its own weight if not precisely engineered. Deploying instruments to the full depth of the Mariana Trench is not a routine operation for most research vessels. Most ships that could attempt it never do. This is the context in which Hadal biology was first attempted. Scientists knew these depths existed. They had measured them in rough outline with echo sounders and weighted lines. They could tell that the deepest points of the ocean were somewhere around 10 to 11,000 meters. What they could not do was go there or send anything there and reliably get it back without extraordinary purpose-built equipment. For most of the 20th century, that equipment did not exist in civilian science. The physics that makes Hadal exploration so difficult begins with pressure, and pressure begins with water's own weight. Water is approximately 800 times denser than air. When you descend through seawater, the weight of the water above you increases with every meter. At the surface, the pressure is 1 atmosphere, the weight of the entire column of air above you, pressing down from the edge of the atmosphere. Every 10 meters of seawater you descend adds 1 more atmosphere of pressure. At 100 meters, a typical recreational scuba dive, the pressure is about 11 atmospheres. At 1,000 meters, the depth of a typical deep water fishery, it is about 100 atmospheres, 100 times the pressure at sea level. At the bottom of the Mariana Trench, at roughly 10,900 meters, the pressure is approximately 1,100 atmospheres. 1,100 atmospheres is not an abstraction. It is a physical force of about 11,000 newtons per square centimeter. To visualize this, consider that a standard car tire is inflated to about 2 to 3 atmospheres. 1,100 atmospheres is the equivalent of stacking approximately 50 large commercial aircraft, total weight of several 100,000 kilograms, on a surface the size of your hand. This force acts uniformly in all directions on any object at that depth. It is not a force that pushes from one side. It is a force that compresses from every direction simultaneously, acting on every surface, every internal volume, every interface between materials of different densities. The implications for engineering are comprehensive. Any sealed airspace, a hollow in a piece of metal, a void in a cable, the interior of an electronic housing will collapse under this pressure unless the walls surrounding it are thick enough to resist the force. Ordinary glass, unless very thick and precisely shaped, shatters. Ordinary plastics compress and deform. Even syntactic foam, the hollow glass microsphere composite used for buoyancy in deep-sea equipment, must be specially formulated to resist compression at Hadal pressures. Glass that works perfectly at 3,000 meters loses its structural integrity at 10,000. The engineering requirements for a vehicle or instrument that must survive at full ocean depth are categorically different from those for equipment designed for the mesopelagic or even the upper abyssal. Titanium is the metal of choice for pressure housings in deep-sea equipment. It has a high strength-to-weight ratio, it is corrosion-resistant in seawater over long periods, and it can be machined with the precision required for pressure-rated housings. Even titanium, though, must be forged and machined with extraordinary care to avoid internal voids or stress concentrations that would fail at depth. Every seal must be carefully designed. Every penetration through a pressure housing, for a cable, a sensor, a viewport, is a potential point of failure. The engineering of a vehicle for full ocean depth is not merely a matter of making things stronger. It is a matter of eliminating every possible failure mode across thousands of components, any one of which could cause a catastrophic implosion under pressure. For biological organisms, the challenge is different in kind, but similar in principle. Life at the cellular level depends on molecules maintaining their shapes and their interactions with one another. Proteins, the enzymes, structural components, and signaling molecules that make metabolism possible, have three-dimensional structures that are precisely matched to their functions. A protein folds into a specific shape because of the balance of chemical forces within and around it. Change the pressure, and you change the balance of those forces. At very high pressures, proteins that evolved at surface conditions will unfold or misfold, losing their biological function entirely. Enzymes stop catalyzing their reactions. Structural proteins lose their mechanical properties. The cellular machinery of an organism adapted to 1 atmosphere of pressure simply does not work at 1,100 atmospheres. Lipid membranes, the thin, flexible films of fatty molecules that form the boundary of every cell and every organelle within a cell, also change their behavior under pressure. At surface conditions, a biological membrane is a fluid mosaic, a 2-layered sheet of lipid molecules that can flex and flow while maintaining a barrier between the cell's interior and its environment. Under high pressure, lipid membranes become more rigid, more ordered, less fluid. A membrane that has become too rigid cannot perform the transport and signaling functions that cells depend on. The cell freezes up, unable to move molecules in and out of its interior. This is the problem that organisms living in the Hadal zone have had to solve through evolution. They cannot change the pressure. They cannot leave. They can only alter their own chemistry to function within the pressure that exists. The solutions they have evolved are not obvious from surface-level biology. and many of them were not discovered until researchers began systematically studying the biochemistry of deep-sea organisms collected by sampling equipment capable of reaching Hadal depths. The most important of these adaptations involves a class of molecules called piezolites, a term derived from the Greek word for pressure. Piezolites are small organic molecules that organisms accumulate in their cells in response to high pressure. They protect proteins and membranes from pressure-induced damage by altering the chemical environment around those structures in ways that counteract the pressure-induced changes in molecular behavior. The most studied and most important of the piezolites in marine organisms is a compound called trimethylamine N oxide, abbreviated TMAO. TMAO is a simple molecule, a nitrogen atom with 3 methyl groups attached, plus 1 oxygen atom. It is water-soluble and can be accumulated in cells to high concentrations. It is also the compound primarily responsible for the characteristic smell of marine fish. When fish die and begin to decompose, bacteria convert TMAO to trimethylamine, which has a strong and recognizable odor. In living deep-sea fish, TMAO is not merely a metabolic byproduct. It is a functional molecule that stabilizes proteins under pressure. The mechanism involves TMAO's interaction with water molecules surrounding a protein. Proteins fold into their functional shapes partly because folding exposes their hydrophobic, water-avoiding regions to less water, while leaving their hydrophilic regions in contact with the aqueous environment. Pressure tends to disrupt this balance by forcing water molecules into spaces that hydrophobic protein regions would prefer to avoid. TMAO counteracts this by making the water around a protein slightly less willing to penetrate those spaces. The net effect is that proteins in high TMAO environments resist pressure-induced unfolding more effectively than proteins in low TMAO environments. In 2014, a study led by Paul Yancey at Whitman College in Washington State, working with colleagues at several institutions, published data showing something remarkable. When they measured TMAO concentrations in marine fish from a range of depths, they found a strict linear relationship between depth and TMAO concentration. The deeper a fish lives, the more TMAO it accumulates, and the relationship is nearly perfectly linear across a range from near-surface fish at essentially 0 TMAO to deep-sea fish at depths of several thousand meters with TMAO concentrations of several hundred millimoles per kilogram of tissue. The fish are not accumulating TMAO arbitrarily. They are calibrating their TMAO levels precisely to the pressure at the depth they live. This finding has implications for how deep fish can go. If TMAO concentration increases linearly with depth, there should be some maximum depth at which the TMAO concentration required for pressure adaptation would be toxic. Too much of it in a cell becomes harmful in its own right. Calculations based on the measured linear relationship suggested that this limit falls somewhere around 8,000 to 8,500 meters, which aligns remarkably well with the observed depth limit of fish in the ocean. No fish has ever been found at depths substantially below the record, which currently stands at 8,136 meters. The biochemistry of TMAO appears to define the biological ceiling on fish depth. Beyond that ceiling, the chemistry of pressure adaptation becomes self-defeating. The story of how humans first reached the bottom of the ocean is one of physics and engineering applied under conditions of profound uncertainty. It involves a vehicle whose design principles seem almost incomprehensibly simple given the environment it was intended to enter. The bathyscaphe Trieste was designed by the Swiss physicist Auguste Piccard and built in Italy in the early 1950s. The name Trieste reflects the city where it was completed. Its design was based on the same principle as a hot air balloon, a float containing a buoyant medium. In this case, aviation gasoline rather than hot air, chosen because gasoline is less dense than seawater and does not compress significantly at great depth, with a heavy observation sphere attached below. The gasoline in the float provided the buoyancy to lift the vehicle. Iron pellets held in hoppers by electromagnets provided ballast. To descend, the electromagnets were switched off and iron pellets released, reducing buoyancy. To ascend, the electromagnets were switched on and iron pellets were shed, allowing the gasoline's buoyancy to lift the vehicle. The observation sphere was the part in which the crew rode. It was a steel sphere with walls nearly 4 inches thick, designed to resist the crushing pressure of the full ocean depth. It was connected to the gasoline float by structural members that transferred the load without creating stress concentrations. The sphere had one window. a tapered cone of plexiglass, wider on the outside than the inside, so that pressure actually sealed it more tightly against the hull rather than forcing it inward. This was a critical design detail. At 1100 atmospheres, any window that pressure could force open would fail catastrophically. The cone design used pressure as part of the seal. The Trieste was purchased by the United States Navy in 1958. In early 1960, it was prepared for an attempt to reach the bottom of the Challenger Deep, the deepest known point in the Mariana Trench. The crew would be Jacques Piccard, son of the designer who had participated in earlier test dives, and Don Walsh, a lieutenant in the United States Navy. On January 23, 1960, they entered the sphere and the hatches were sealed. The descent to the bottom of the Challenger Deep took nearly 5 hours. The Trieste moved through the water at roughly a meter per second, sometimes slightly faster. During the descent, the plexiglass window cracked. Not catastrophically, not enough to compromise the integrity of the sphere, but enough that the two men inside heard the crack and felt the vibration of its propagation through the hull. They continued. There was no radio communication with the surface during the descent. The only sound was the hum of instrumentation and the creak of the structure settling under increasing pressure. When the Trieste touched the bottom, sonar instruments showed a depth of approximately 10,916 meters. The landing sent a cloud of fine white diatomaceous sediment billowing upward from the floor, obscuring the view through the single window. Piccard and Walsh spent approximately 20 minutes on the bottom, waiting for the sediment to settle. During that time, Piccard believed he saw a flatfish, something with the shape of a flounder or sole, resting on the sediment near the vehicle. Subsequent marine biologists have questioned this identification, suggesting the animal may have been a sea cucumber or some other organism seen imperfectly through the disturbed sediment and the limited window. The identification was never confirmed. What Piccard and Walsh did confirm was something more fundamental. It was possible for a vehicle containing human beings to reach the bottom of the Mariana Trench and return safely. They then ascended, a process that took another 3+ hours. When they surfaced, they had visited the deepest point on Earth. No human being would return for 52 years. That interval, from January 1960 to March 2012, represents one of the most striking gaps in the history of exploration. During those 52 years, 12 human beings walked on the surface of the Moon. Robotic spacecraft flew past every planet in the solar system. Human beings launched and maintained orbital stations continuously. The surface of Mars was mapped at better resolution than the bottom of the Mariana Trench. The human genome was sequenced, the internet was built, and the deepest point on Earth was visited by no one. The Challenger Deep was probed occasionally by unmanned vehicles and instrument packages. In 1995, a Japanese remote-operated vehicle called Kaiko reached the Challenger Deep and returned video footage of the sediment surface. In 2009, a hybrid remotely operated vehicle called Nereus reached the Challenger Deep as well. But no human returned until James Cameron. Cameron's dive in March 2012 was undertaken in a vehicle called the Deep Sea Challenger, a purpose-built submersible designed specifically for the attempt. Unlike the Trieste, which had been a large vehicle requiring a support ship with heavy lift capacity, the Deep Sea Challenger was relatively compact and designed to descend faster. Cameron piloted it alone. The descent took approximately 2.5 hours, reaching the bottom in less time than the Trieste had managed decades earlier. Cameron spent approximately 3 hours on the bottom, operating cameras and collecting sediment samples. He brought back footage showing a pale, flat, nearly featureless landscape of soft sediment. What he described as a desolate place, surprisingly quiet, without the complex biological communities he had half expected to see on the basis of analogy with hydrothermal vent environments. The Deep Sea Challenger had several technical problems during the dive. Hydraulic fluid leaked, limiting the mobility of the vehicle's sample collection arm. The thruster system did not perform entirely as intended. Cameron described the vehicle as less capable in operation than it had been in testing. He collected samples, operated cameras, and returned to the surface. The scientific return was meaningful but limited by the mechanical difficulties. The vehicle was not used again for Hadal exploration after this mission. The systematic effort that changed Hadal science most profoundly came later, between 2018 and 2019, when Victor Vescovo undertook what he called the 5 Deeps expedition. Vescovo was a retired United States Navy officer and private investor who had developed a sustained interest in deep ocean exploration. The Five Deeps project set out to visit the deepest point in each of the five oceans: Atlantic, Southern, Indian, Pacific, and Arctic, and to do so with a vehicle robust enough to make multiple dives at each location. The vehicle Vescovo used was called the Limiting Factor, built by Triton Submarines in the United Kingdom. Unlike the Trieste, which relied on gasoline buoyancy, or the Deep-Sea Challenger, which used a novel foam and sphere design, the Limiting Factor was a modern pressure vessel built entirely from titanium, equipped with advanced battery systems, high-definition cameras, and sampling equipment. Its pressure sphere was designed to withstand full ocean depth, defined as any point down to 11,000 meters. Not once, but repeatedly. The vehicle was tested at full pressure before any deep dives were attempted. The Five Deeps expedition began in late 2018 with a dive to the Puerto Rico Trench, the deepest point in the Atlantic Ocean, at approximately 8,376 meters below sea level. From there, the expedition moved to the Southern Ocean and the South Sandwich Trench, reaching approximately 7,432 meters at a point called the Factorian Deep, then to the Indian Ocean and the Java Trench, also called the Sunda Trench, where the deepest point reaches approximately 7,187 meters. Then to the Pacific and the Challenger Deep in the Mariana Trench, where Vescovo's instruments recorded a depth of approximately 10,928 meters, slightly deeper than previous measurements, suggesting either a refinement of the measurement or a previously unrecorded local low point. Finally, to the Arctic Ocean and the Malloy Deep, at approximately 5,551 meters. Each of these dives added to the photographic and biological record of the Hadal Zone. At the Puerto Rico Trench, Vescovo observed and collected samples of sea cucumbers and amphipods. At the Challenger Deep, he found something that received considerable attention in the scientific and popular press: plastic debris on the sediment surface. A plastic bag was visible in footage from the dive. Plastic had reached the deepest point on Earth before any systematic attempt had been made to survey the biological communities there. This was not a surprise to Hadal biologists. The contamination studies had already suggested it was likely. But seeing it visually in footage from the bottom crystallized the reality in a way that data tables and journal papers cannot. The total cost of the Five Deeps expedition was approximately $48 million. This included the construction of the Limiting Factor, the purchase and outfitting of a support ship, the salaries of crew and scientific personnel across multiple expeditions lasting months, and the operational costs of running a complex deep-sea research program across 5 oceans over more than a year. $48 million is a significant sum in any context. In the context of ocean exploration funding, it represents roughly the total annual oceanographic research budget of a mid-sized research institution, spent on one project over 1 year to visit 5 locations. When scientists and engineers began systematically visiting the Hadal Zone with cameras and sampling equipment, what they found was not what the abyssal plain had led them to expect. The abyssal plain, the flat, sediment-covered terrain that makes up most of the ocean floor between roughly 3,000 and 6,000 meters, is a relatively sparse biological environment. Food arrives slowly, in the form of marine snow drifting down from the productive surface waters above, and the community of organisms that lives there is calibrated to this slow, dilute food supply. The Hadal Zone, deeper by definition, ought to be even sparser, less food arriving, more pressure to cope with, colder temperatures. The expectation would be a biological community thinner and simpler than the abyssal. The reality is more complex, and in some respects opposite to this expectation. The fauna of the Hadal Zone is distinctive, not a subset of abyssal fauna living at greater depth, but a qualitatively different community, with its own characteristic species, its own food web structure, and its own unexplained patterns of abundance and diversity. The trenches are not barren. They are, in a sense, enriched, concentrated ecosystems supported by the geological structure of the trench itself. Understanding why requires understanding what lives there. The most visible and most remarked upon organisms in the Hadal Zone are the snailfish. Snailfish belong to the family Leparidae and are found throughout the ocean from shallow coastal waters to the deepest trenches. The deep-sea species are remarkable creatures that have been filmed, described, and studied with increasing detail as camera technology capable of reaching Hadal depth has improved. They are the dominant fish fauna of the trenches, not merely present but abundant, active, and apparently well-fed. In 2014 and subsequent years, camera landers deployed to the Mariana Trench returned footage of snailfish in numbers that surprised researchers. Rather than the occasional solitary fish expected in such a food-poor environment, the footage showed groups of 5, 10, sometimes more than 20 individuals swarming the bait used to attract organisms to the camera. The fish moved quickly and competed aggressively for food. They were pale, almost translucent, with a gelatinous body texture and no obvious external armor. They looked, to an untrained eye, like extremely large, very pale tadpoles. This appearance reflects a genuine biological adaptation. The snailfish's gelatinous body structure is an advantage at depth rather than a vulnerability. Most fish have a swim bladder, a gas-filled internal sac used for buoyancy control. At Hadal pressures, a gas-filled swim bladder would be crushed. Snailfish at Hadal depth have no functional swim bladder. Their bodies are largely water, with a musculoskeletal system embedded in a gelatinous matrix that is essentially incompressible. Because water cannot be compressed at the pressures encountered in the Hadal zone, at least not to any significant degree, a body built primarily of water has no internal volume that pressure can collapse. The fish are already adapted to pressure simply by virtue of being mostly liquid. Pseudoliparis swirei, the species most associated with the Mariana Trench, was formally described by Thomas Linley and colleagues in a paper published in 2017, based on specimens collected from depths between 6,898 and 7,966 meters. The description was the culmination of years of camera observations and the first physical collection of specimens from that depth in that trench. The species was named for Herbert Swire, the ship's navigator on the HMS Challenger during its 1872 to 1876 expedition, the expedition that first measured a deep point in the Mariana Trench. The naming was a deliberate homage to the long chain of observation that connects the first rough soundings to the detailed biological description more than a century later. In 2023, a new depth record for fish observation was set. A lander deployed in the Izu-Ogasawara Trench, which runs south from Japan, parallel to the arc of the Izu and Ogasawara island chains, returned footage of a juvenile snailfish at a depth of 8,236 meters. This individual was later identified as Pseudoliparis belyaevi, a species known from shallower records in the same trench system. The footage was captured by a lander operated jointly by researchers from the University of Western Australia and the Tokyo University of Marine Science and Technology, part of a broader survey effort targeting Hadal fish communities across the Pacific Trench systems. The record depth of 8,336 meters is, as noted earlier, very close to the theoretical maximum depth predicted by the TMAO biochemistry calculations, a coincidence that is probably not a coincidence at all. Alongside the snailfish, the megafaunal ecology of the Hadal zone is dominated by crustaceans, specifically by the Lysianosoid amphipods that swarm in extraordinary densities around any source of organic material in the trenches. Amphipods are small, laterally compressed crustaceans found throughout the ocean and in many freshwater environments. In the Hadal zone, they have radiated into a diversity of forms adapted to the extreme conditions, and their abundance is often strikingly high given the apparent scarcity of food. Hirundellea gigas is the dominant amphipod species in the Mariana Trench and is found throughout the deeper parts of the Pacific Hadal zone. It is a relatively large amphipod by the standards of the group, reaching lengths of 2 to 3 centimeters, and it is an opportunistic scavenger of extraordinary effectiveness. When bait is placed on the trench floor by a lander, Hirundellea gigas typically arrives within minutes to hours, faster than any other large organism, and can strip a fish carcass to bare bones in a matter of hours with the right aggregation size. Its speed of arrival suggests that individual amphipods travel substantial distances across the trench floor in search of food, using chemical gradients in the water to locate organic material far away. One of the most unusual aspects of Hirundellea gigas biology is its ability to digest cellulose, plant material, using gut enzymes that most animals do not produce. The amphipods have been found containing fragments of wood in their digestive tracts, and biochemical analysis has confirmed the presence of cellulose enzymes in their gut tissue. This capability allows them to exploit wood and plant debris that sinks to Hadal depth after being carried out to sea by rivers and ocean currents, a food source that almost no other deep-sea scavenger can access. In other trench systems, particularly the Kermadec Trench, east of New Zealand, researchers have found amphipods belonging to the species Allicella gigantea, which represent the opposite extreme of Hadal amphipod size. Allicella gigantea can reach lengths of up to 34 centimeters, roughly the length of a forearm, making it the largest known amphipod in the ocean. Its size is an example of what biologists call deep-sea gigantism, a pattern observed in multiple taxonomic groups in which organisms from extreme deep-sea environments are substantially larger than their relatives from shallow or mid-water environments. The mechanisms behind this gigantism are debated. Leading explanations include the scarcity of food, larger body size allows storage of more energy reserves between meals, and the reduced predation pressure in an environment where only a limited range of predators exists. Larger individuals may also be better competitors for the rare large food falls that represent the most significant input of energy to the Hadal community. The megafaunal community of the Hadal zone also includes holothurians, sea cucumbers, adapted to function at extreme pressure and temperature. Holothurians are echinoderms, relatives of sea urchins and sea stars, and they are one of the most ubiquitous groups of animals in the deep sea generally. Hadal holothurians sift sediment for organic particles, moving slowly across the trench floor and extracting nutrition from the thin veneer of organic carbon deposited on the sediment surface. Some species have been filmed swimming in the water column above the trench floor, propelling themselves with undulating body movements in a manner unusual for a group more typically associated with slow benthic movement. Polychaete worms form another significant component of the Hadal fauna, both at the sediment surface and within the sediment itself. Some polychaetes are predators, using elaborate jaw structures to capture other invertebrates. Others are deposit feeders, ingesting sediment and extracting organic material from it as it passes through their digestive system. Others still are suspension feeders, using feathery or bristled appendages to capture particles from the water column. The diversity of feeding strategies within a single animal group at these depths reflects the general principle that evolution will fill available ecological niches even in the most extreme environments, that where there is any variation in how food can be obtained, organisms will eventually specialize to exploit that variation. Single-celled organisms, foraminifera, which build elaborate shells of calcium carbonate or agglutinated sediment particles, and xenophyophores, which are giant single cells that can reach plate-sized dimensions, are also present at Hadal depths. Xenophyophores at Hadal depth are among the largest single cells ever documented, their extraordinary size made possible partly by the cold temperature and the physical support provided by their agglutinated test. They sit on the sediment surface, collecting particles from the water above, and their presence suggests that even single-celled organisms have found ways to exploit the Hadal environment productively. The communities of the Hadal zone are not simply abyssal communities under greater pressure. They are distinct assemblages with their own characteristic dominant taxa, their own food web structure, and, in many cases, their own endemic species found nowhere else in the ocean. Two adjacent trenches may share some species and differ significantly in others, depending on the distance between them, the depth of the sills separating them from each other and from the surrounding abyssal plain, and the local history of colonization and extinction events. Understanding this biogeography, the geographical distribution of species across Hadal environments, requires sampling from multiple trenches at comparable depths and comparing what is found. This work has barely begun. The lander solution emerged from necessity. If crewed vehicles were too expensive and too rare to deploy systematically, and if tethered ROVs were limited by the mechanical complexity of managing cables across 11,000 meters of water column, then what remained was an instrument that could reach the bottom on its own, do its work, and come back on its own. This is the lander, a weighted frame of metal and syntactic foam carrying cameras, sensors, and baited traps that free falls to the Hadal floor and returns by passive buoyancy once its ballast is released. The mechanics of a deep-sea lander are straightforward in principle and demanding in practice. The basic design is a framework, typically stainless steel or aluminum, to which all the instrument packages are attached. Weight is added at the bottom in the form of iron plates or concrete blocks, enough to ensure that the lander sinks at a predictable rate despite the buoyancy of the foam and instrument housings. The baited trap, usually a wire mesh or plastic bottle arrangement containing fish or squid, is positioned where it can be filmed by the cameras. The cameras themselves run on battery power, recording continuously or at intervals depending on the mission duration. Lights are attached because the Hadal zone has no illumination whatsoever, and any organism captured on film must be illuminated by the lander's own light source. When the time comes to return to the surface, the lander does not winch itself up. There is no motor or cable involved. Instead, the ballast is released by an acoustic command from the ship above, or by a mechanical timer set before deployment. The acoustic release mechanism consists of a transponder mounted on the lander that listens for a specific acoustic signal from the ship's transducer. When it receives the right frequency and code, it triggers a mechanism that allows the ballast weights to drop free. The syntactic foam, which has been supporting the weight of the ballast during the descent and bottom time, then lifts the lander slowly toward the surface. This process takes hours in either direction. A lander deployed at 11,000 meters depth will spend approximately 5 hours sinking, some number of hours sitting on the bottom, typically between 4 and 24 hours depending on the mission, and then another 5 hours rising. The ship must maintain position in the deployment area for the full duration, waiting for the lander to surface. When it does, it appears as a small floating object in a very large ocean, and the crew must locate it using a radio beacon or visual signal before it can be recovered. The elegance of the lander is that it can be built and deployed at a fraction of the cost of a remotely operated vehicle. A basic deep-sea lander can be constructed for tens of thousands of dollars rather than millions. It requires no umbilical cable, no real-time telemetry, no complex maneuvering system. Its limitations are equally straightforward. It goes where you drop it and stays where it lands. It cannot pursue an organism that moves. It cannot sample a specific feature of the seafloor that is 10 meters from where it happened to land. It cannot collect a delicate specimen without crushing it in a trap. The lander provides a persistent presence in a place where any presence is remarkable, but it provides that presence at the price of mobility. The Hadal Ecosystem Studies Project, known as HADES, was one of the first systematic efforts to use landers to build a comprehensive picture of biological communities across multiple Hadal trenches simultaneously. The project was led by Jeff Drazen, a deep-sea biologist at the University of Hawaii at Manoa, and Patricia Fryer, a marine geologist at the same institution. It was funded by the National Science Foundation, beginning in 2014. The HADES project deployed landers across both the Mariana Trench and the Kermadec Trench, collecting parallel datasets from two of the major Pacific Hadal systems, allowing the first meaningful comparative analysis of how Hadal biology varies between different trench environments. What the HADES project found reinforced and extended what previous camera surveys had suggested. The Mariana and Kermadec trenches had different biological communities despite both being deep Pacific Hadal environments. The Kermadec Trench, located northeast of New Zealand, had higher abundances of certain amphipod species and different fish assemblages than the Mariana Trench. The differences reflected the geographic isolation of each trench, separated from the other by thousands of kilometers of abyssal plain that Hadal organisms cannot cross without passing through shallower water, as well as differences in the overlying productivity of the surface ocean, which determines how much organic material sinks to the Hadal floor. The HADES project data contributed to understanding the geological amplifier effect, the observation that Hadal trenches concentrate organic material from a much wider area than the trench floor itself. Material on the slopes surrounding a trench does not stay on those slopes. Gravity, earthquakes, and the occasional submarine landslide carry material from the slope downward and into the trench axis. The trench acts as a collecting basin for organic material from a large catchment area. The result is that the actual flux of organic material arriving at the floor of a Hadal trench can be substantially higher than would be expected simply from the productivity of the surface waters directly overhead. This is why trench floors can support higher organism densities than comparable abyssal plains, despite being deeper and nominally more food-limited. The contamination findings that emerged from Hadal research in the 2010s were as significant as any biological discovery from the same period, and more troubling in their implications. The discovery that persistent organic pollutants had reached the deepest points in the ocean was not entirely unexpected. Scientists studying marine pollution had long suspected that the slow rain of particles carrying these compounds would eventually carry them to any depth, but the concentrations found were much higher than had been modeled. Alan Jameson is a marine biologist who has dedicated much of his career to the development and deployment of deep-sea landers, and to the analysis of the organisms collected by those landers. Working at the time at Newcastle University in the United Kingdom, Jameson led a study that collected amphipods from two of the deepest trench systems in the Pacific, the Mariana Trench at approximately 10,190 meters and the Kermadec Trench at approximately 9,000 meters. The amphipods were Hirundellea gigas and other Lycianosoid species. The samples were analyzed for two classes of persistent organic pollutants, polychlorinated biphenyls, known as PCBs, and polybrominated diphenyl ethers, known as PBDEs. PCBs were manufactured industrially from the late 1920s until the 1970s and 1980s, when they were banned in most countries because of their toxicity and their tendency to persist in the environment. They were used primarily as dielectric fluids in electrical transformers and capacitors, as hydraulic fluids, and as plasticizers. Their chemical stability, the property that made them useful industrially, also makes them resistant to biological degradation. They accumulate in fatty tissue and concentrate through food chains. By the time they were banned, vast quantities had already been produced and released into the environment, and the amount that had entered the global ocean system was enormous. PBDEs were manufactured as flame retardants and used widely in furniture foam, electronics, and building materials. Like PCBs, they are chemically stable, fat-soluble, and resistant to biodegradation. They were used extensively beginning in the 1970s and continued in production in many countries until regulatory pressure in the 2000s and 2010s led to bans or restrictions. Both classes of compounds enter the ocean from multiple pathways. Industrial spills, waste disposal, runoff from landfills, degradation of products containing these compounds, all of these release PCBs and PBDEs into coastal waters. From coastal waters, the compounds adsorb to particles, fragments of plastic, dead plankton, fecal pellets, any small piece of organic material that floats in the water. Those particles sink. As they sink, they carry the adsorbed pollutants downward. The particles degrade as they sink, releasing some of the pollutants into solution, but many remain bound to organic matter until they reach the deep sea floor. The concentrations of PCBs found in the Mariana Trench amphipods by Jameson's 2017 study were, on average, around 900 nanograms per gram of lipid. The concentrations of PBDEs were somewhat lower. These numbers require context to be meaningful. For comparison, the Liaohe River in northeastern China, one of the most heavily polluted rivers in the world by organic contaminant load due to decades of industrial activity in its drainage basin, contains PCB concentrations in bottom sediments of roughly 300 to 600 nanograms per gram. The amphipods from the Mariana Trench, at 11,000 meters, in the most remote marine environment on Earth, contained PCB concentrations exceeding those from one of the most polluted rivers on the planet. The Kermadec Trench samples showed even higher values. Average PCB concentrations in Kermadec amphipods were approximately 1,900 nanograms per gram of lipid, more than 3 times the Mariana Trench values, and many times the concentrations in the most polluted coastal sediments measured. The higher values at Kermadec may reflect differences in the geographic proximity to historical PCB sources, differences in the particle transport pathways between the two trench systems, or differences in the local amphipod biology that affects how contaminants accumulate. The pathway that delivers these compounds to the deepest part of the ocean is the same pathway that delivers organic material generally. The slow rain of particles from the surface, concentrated by the trench's geological amplifier effect. PCBs and PBDEs adsorb preferentially to lipid-rich particles, the most calorie-dense and therefore most desirable food items for deep-sea scavengers. The amphipods eat the food that arrives at the trench floor, and with that food, they ingest the contaminants it carries. Because PCBs and PBDEs resist biological degradation, the amphipod's own metabolism cannot break them down. They accumulate in the animal's fatty tissue, building up over the course of its life. Since Jameson's 2017 study, subsequent research has confirmed the presence of microplastics, heavy metals, and additional classes of persistent organic contaminants in Hadal organisms from multiple trench systems. The concept of the Hadal zone as a pristine or remote ecosystem, isolated from human chemical influence, is not supported by the data. The trenches are remote in the sense of being difficult to reach. They are not isolated from the surface ocean's chemistry. The microbiology of the Hadal zone operates at a different scale from the macrofauna, bacteria rather than fish, archaea rather than amphipods, but is no less important to the functioning of the Hadal ecosystem. In terms of biomass, microbial organisms vastly outnumber all the macrofaunal species combined. In terms of chemical cycling, microorganisms perform most of the transformations that determine what nutrients are available to larger organisms and what compounds are returned to the water column as organic matter degrades. Microbiologists classify deep-sea bacteria and archaea into categories based on their response to pressure. Piezo-tolerant organisms are those that can grow at elevated pressure but grow equally well or better at surface pressure. Piezophiles are organisms that grow better at elevated pressure than at surface pressure. Pressure is advantageous rather than merely tolerable for them. Obligate piezophiles are organisms that require elevated pressure to grow and cannot survive at or near surface pressure. The Hadal zone is dominated, in terms of microbial ecology, by piezophilic and obligate piezophilic organisms, creatures for which pressure is not a challenge to be overcome, but a condition of existence. Doug Bartlett at the Scripps Institution of Oceanography in La Jolla, California, has been among the most prominent researchers studying the molecular and physiological basis of piezophily. Bartlett's laboratory has worked extensively with deep-sea bacterial isolates collected from the Pacific trenches, studying how their membrane composition, their enzyme systems, and their gene regulation differ from those of surface bacteria. One of Bartlett's key findings concerns the lipid composition of piezophilic bacterial membranes. In surface bacteria, cell membranes are composed primarily of lipids with saturated or monounsaturated fatty acid chains. These membranes are fluid enough at surface pressure and surface temperature to allow the transport proteins and receptor proteins embedded in them to function. Under high pressure, these standard bacterial membranes become too rigid. The pressure forces the lipid molecules into a more ordered arrangement that reduces fluidity. Piezophilic bacteria compensate for this by incorporating a higher proportion of polyunsaturated fatty acids into their membranes. Polyunsaturated fatty acids have more double bonds in their carbon chains, which prevents the chains from packing tightly and maintains membrane fluidity even under the compressive force of high pressure. Moritella japonica is a piezophilic bacterium isolated from a depth of approximately 6,000 meters in the Japan Trench in the 1990s. It is one of the most studied of the Hadal bacteria, partly because it can be cultured under simulated Hadal pressure conditions in specially built pressure vessels in the laboratory. It grows fastest at approximately 700 atmospheres, roughly corresponding to its natural habitat depth, and grows poorly at surface pressure. Its genome has been sequenced, revealing adaptations across many aspects of its biology, from its membrane lipid synthesis pathways to its protein folding machinery. Shewanella benthica is another piezophilic species found throughout the Hadal zone, recovered from multiple trench systems including the Mariana and Philippine trenches. Shewanella species are facultative anaerobes. They can grow with or without oxygen, and they are important players in the reduction of iron and manganese compounds in deep-sea sediments, a process that cycles these metals through the sediment-water interface and has implications for the chemistry of the overlying water. Beyond the bacteria, the Hadal zone harbors a distinctive archaeal community. Archaea are a domain of life distinct from bacteria, sharing some superficial resemblances with bacteria, but differing fundamentally in the chemistry of their membranes, their gene expression machinery, and many aspects of their metabolism. In the deep ocean, archaea often outnumber bacteria in terms of cell counts in the water column, and they play critical roles in the nitrogen cycle, particularly in the process of ammonia oxidation that converts ammonium ions to nitrite. In the Hadal zone, a lineage of archaea known as the deep-sea archaeal group, or DSAG, is consistently detected in environmental surveys of Hadal sediments. The DSAG is known almost entirely from DNA sequences extracted from environmental samples. Its members have never been successfully cultured in the laboratory. Their metabolic capabilities are therefore inferred from genomic analysis rather than direct observation. Genomic data suggests that DSAG archaea are capable of processing various forms of organic carbon under anaerobic or low oxygen conditions, making them potentially important in the degradation of the organic material that accumulates in trench sediments. The problem of the uncultured majority, the observation that the vast majority of deep-sea microorganisms cannot be grown in laboratory culture, is a fundamental constraint on Hadal microbiology. Standard microbiological methods involve collecting organisms and growing them in culture medium under controlled conditions, then characterizing their physiology and biochemistry. For Hadal microorganisms, this approach fails at multiple points. The organisms require high pressure to survive, which means culture must be conducted in high-pressure vessels, equipment that is expensive, difficult to work with, and limited in the number of culture conditions that can be tested simultaneously. Furthermore, many Hadal microorganisms have nutritional requirements or environmental dependencies that are not replicated by standard culture media formulated for surface microorganisms, The result is that Hadal microbiology has been transformed by culture-independent methods, particularly the sequencing of environmental DNA extracted directly from sediment or water samples. By extracting all the DNA from a sample and sequencing it, researchers can identify the organisms present in a community without ever culturing a single individual. The method is powerful for detecting who is present, but limited in what it can tell us about what those organisms are doing. The gap between detection and understanding is wide in Hadal microbiology, and filling that gap requires methods that are still being developed. The Hadal food web depends ultimately on what falls from above, and the character of what falls is itself a complex story. Marine snow is the term used for the continuous slow rain of organic particles that drifts downward through the ocean from the productive surface waters. The particles are heterogeneous in composition: dead phytoplankton cells, fecal pellets produced by zooplankton feeding in the surface ocean, marine aggregates that form when dissolved organic matter clumps into particles, fragments of larger organisms, and various other forms of organic debris. Each particle begins sinking when it becomes dense enough that its negative buoyancy exceeds any upward force acting on it. The sinking rate of marine snow particles is slow, typically between 1 and several hundred meters per day, depending on particle size and density. A particle leaving the surface may take weeks to reach the abyssal plain at 3,000 to 4,000 meters depth, and weeks more beyond that to reach a Hadal trench at 10,000 to 11,000 meters. During this journey, bacteria colonize the particle and begin degrading it. Each meter of descent removes some fraction of the particle's organic content as bacteria consume and respire the organic carbon. By the time the particle reaches Hadal depth, it has been degraded substantially, and what arrives at the trench floor is a shadow of what left the surface. The quantity of organic material that reaches the Hadal floor from marine snow alone is difficult to measure, but is known to be small compared to the flux at shallower depths. Estimates of organic carbon flux at Hadal depths suggest values on the order of a few milligrams of carbon per square meter per day, amounts that would seem insufficient to support the amphipod abundances and snailfish populations that cameras have documented. The geological amplifier effect, the concentration of organic material by the trench's geometry and by mass wasting events from the trench walls, partially resolves this discrepancy. But the full accounting of energy input to the Hadal ecosystem remains uncertain, and some researchers suspect that chemolithotrophy, the use of chemical energy from inorganic compounds rather than from organic carbon, plays a role in some trench systems. Cold seeps at Hadal depths, areas where methane, hydrogen sulfide, or other reduced compounds seep from the sediment through tectonic fault structures, would support chemolithotrophic bacteria that could form the base of a locally sustained food web, independent of the rain of organic material from above. Evidence for cold seep activity has been found in some trench systems, and the associated biological communities in shallower seeps are well documented. Whether equivalent communities exist at Hadal depth in all trench systems or only in some, remains an open question. Whale falls represent a sporadically but significantly productive food input to the Hadal zone. A blue whale carcass, the largest animal body available to the deep ocean floor, weighs perhaps 90,000 kilograms. When a blue whale dies in the ocean, it sinks over a period of days to weeks, depending on the state of decomposition and the depth of water. Carcasses sinking in areas directly above Hadal trenches may eventually reach trench depth, delivered as an enormous packet of organic material to an ecosystem whose annual organic carbon input from marine snow alone might be measured in grams per square meter. The biology of whale falls has been studied in detail at abyssal depths. The community succession from mobile scavengers stripping the flesh to sulfophilic bacteria colonizing the lipid-rich bones is now reasonably well characterized. At Hadal depth, the whale fall community would presumably differ in its species composition from abyssal whale fall communities, given the different fauna available, but the ecological function would be similar. The evidence for whale falls at Hadal depth is indirect. Bone fragments have been recovered in Hadal sediment samples, but it is consistent with the expectation that carcasses from areas of high whale density in the overlying waters would occasionally reach even the deepest trenches. The Tonga Trench, located in the southwestern Pacific between the Tonga Islands and the Fiji Basin, is the second deepest oceanic trench system on Earth. Its maximum depth, the horizon deep, reaches approximately 10,882 meters, deep enough to submerge Mount Everest with still more than a kilometer of water above its summit. The Tonga Trench marks the boundary between the Pacific and Australian tectonic plates and is one of the most seismically active boundaries on the planet. The rate of plate convergence at the Tonga subduction zone is among the fastest measured anywhere on Earth, generating frequent earthquakes and the volcanic arc of the Tonga Islands. The biological community of the Tonga Trench differs from that of the Mariana Trench, despite comparable depths. The amphipod fauna shows a different species composition. Different lysianosoid species dominate the two systems, and the fish community, while similarly dominated by snailfish, shows differences in species present and their abundance relative to depth. These differences reflect the geographic isolation of the two trenches from each other and their different histories of colonization. The Tonga Trench is separated from the Mariana Trench by the shallow seamount chains and island arcs of the Central Pacific, which form barriers that prevent hadal species from crossing between the two systems without passing through shallower water that they cannot survive. The Kermadec Trench extends southwest from the Tonga Trench and reaches depths of approximately 10,047 meters. It is in the Kermadec Trench that some of the most detailed lander-based surveys of hadal biology have been conducted, including the Hades Project surveys mentioned earlier. The Kermadec system is notable for its high abundances of Alicella gigantea, the giant amphipod that can reach 34 centimeters in length, and for the diversity of its polychaete worm community. Environmental DNA surveys of Kermadec water and sediment samples have revealed genetic signatures of many organisms not captured in physical traps, suggesting that the biological diversity of this trench system exceeds what camera and trap surveys have documented. The Japan Trench, which runs along the eastern coast of Japan's main islands at depths reaching approximately 9,000 meters, became an unusually dynamic research environment after the Tohoku earthquake and tsunami of March 2011. The earthquake, with a moment magnitude of 9.0, was one of the largest ever recorded. It occurred along the Japan subduction zone, where the Pacific Plate is descending beneath the North American and Okhotsk plates. The ground motion at the sea floor was violent enough to trigger massive submarine landslides on the slopes of the Japan Trench. The Japan Agency for Marine Earth Science and Technology, JAMSTEC, operates the Shinkai 6500, a research submersible capable of diving to 6,500 meters. The Shinkai 6500 had conducted surveys of the Japan Trench before 2011 and returned to the area after the earthquake to document the changes to the seafloor. The before and after comparison was striking. Entire sections of the slope had collapsed, depositing material on the trench floor that had not been there previously. The sediment surface in areas that had been relatively stable showed extensive disturbance. In some areas, the previous biological communities, the organisms that had been colonizing the sediment over years or decades, had been buried under meters of freshly deposited landslide material. This kind of disturbance is not merely a curiosity. It raises fundamental questions about the nature of biological communities at hadal depth. If a trench floor community can be largely destroyed by a single seismic event and must subsequently recolonize from scratch, amphipods migrating in from less disturbed areas, polychaete worms recolonizing from surviving populations, then the observed communities at any given time reflect not only the stable physical environment, but also the history of disturbance events that have affected that location. Two surveys of the same location separated by a decade might find entirely different communities if a major earthquake occurred in the interval. The ecological dynamics of hadal communities are therefore closely tied to the geological dynamics of the subduction zones that create the trenches. The Puerto Rico Trench is the deepest point in the Atlantic Ocean, reaching approximately 8,376 meters at a location called the Milwaukee Deep. It runs along the boundary between the Caribbean and North American plates, northwest of Puerto Rico, and is the result of a complex tectonic setting that differs substantially from the simple subduction geometry of the Pacific trenches. The Atlantic is geologically quieter than the Pacific. There are no Ring of Fire equivalent volcanic arcs along its margins, and its hadal environment is correspondingly more stable, and, at a maximum depth of 8,376 meters, shallower than the deepest Pacific trenches. The biology of the Puerto Rico Trench has been less extensively studied than that of the major Pacific trenches, partly because the trench is shallower and therefore attracts less attention from researchers focused on absolute depth records, and partly because the Atlantic has fewer institutions with deep-sea research programs than the Pacific margins. What data exists suggests that the amphipod community of the Puerto Rico Trench is distinct from Pacific hadal amphipod assemblages reflecting millions of years of independent evolution in geographic isolation. Fish surveys have found snailfish present, as expected, but at lower relative abundances than in equivalent Pacific depths. The approach of using environmental DNA to survey biological communities was first applied to shallow marine environments and has been progressively extended to deeper and more difficult environments as sampling and sequencing technology has improved. At Hadal depths, eDNA sampling involves collecting water samples, typically tens to hundreds of liters, during a lander deployment or ROV dive, filtering them through fine membranes that capture any cellular material or shed DNA in the water, and then sequencing the genetic material on the filters. The resulting data contains sequences from every organism that has shed DNA into the water near the sampling location, including organisms that were never physically captured. The limitations of eDNA surveys at Hadal depth are multiple. The water column above the trench floor is enormous, and DNA shed by organisms at midwater depths can drift downward on sinking particles to reach the trench floor, where it might be detected and incorrectly attributed to Hadal organisms. The identification of organisms from eDNA sequences is only as good as the reference databases available, and for many deep-sea taxa, no reference sequence exists, making identification impossible below the family or order level. And the relationship between eDNA concentration and organism abundance is non-linear and difficult to calibrate, making eDNA surveys better for presence-absence data than for quantitative estimates of biomass or population size. Despite these limitations, eDNA surveys of the Hadal zone have repeatedly detected genetic signatures of organisms that were not captured by traps and not observed on cameras. Some of these signatures correspond to known deep-sea taxa at unexpected depths. Others correspond to sequences that cannot be matched to any described species and may represent undescribed organisms. The consistent message from these surveys is that the biological inventory of Hadal trenches, as assembled from physical sampling and camera observation, is substantially incomplete. Communication with vehicles and instruments at Hadal depth is constrained by physics in ways that have no easy engineering solution. In shallow water, divers can communicate with the surface by radio from a few meters depth. Remotely operated vehicles in the upper few thousand meters can in principle be guided by acoustic telemetry with reasonable response times. But at 10,000 meters, the round-trip travel time for a sound pulse is approximately 13 seconds. 6.5 seconds down, 6.5 seconds up. Any command sent to a vehicle takes that long to arrive. Any response takes that long to return. If an operator on a ship sends a command at time 0, they will not know whether the vehicle received it until 13 seconds later, and will not see the response to any action taken in response to that command for another 13 seconds after that. This latency is fundamentally different from the experience of operating a drone or remotely controlled robot in air or in shallow water, where response times are measured in milliseconds. At Hadal depth, the interaction between a human operator and a remote vehicle is necessarily slow and discontinuous. An operator who tries to control a vehicle in real time will find themselves always reacting to the vehicle's state 13 seconds in the past. If the vehicle encounters an obstacle or a point of interest, by the time the operator sees it on the acoustic or visual telemetry and sends a response, the vehicle has already moved past it. The practical consequence is that vehicles at Hadal depth must operate semi-autonomously or fully autonomously, following pre-programmed survey paths and making their own decisions about how to respond to immediate environmental conditions. This is not merely an inconvenience. It fundamentally limits what Hadal exploration can accomplish. A marine biologist watching footage of a deep-sea creature through a viewport or video feed can, in shallow water or at abyssal depths where tether latency is manageable, direct the vehicle to follow the organism, adjust the camera angle, deploy a collection arm, take a sample. At Hadal depth, that same biologist can only watch footage that arrives 13 seconds out of date and issue commands that arrive 13 seconds in the future. The creature is gone before the command to follow it is received. The economics of Hadal research constrain it further. The limiting factor, the vehicle used in the 5 Deeps expedition, was built at a cost of approximately $12 million. The support ship needed to operate it cost additional tens of millions to purchase and outfit. The operational costs of a research expedition using the limiting factor, fuel, personnel salaries, scientific equipment, port fees, insurance, run to several $100,000 per day at sea. For a typical academic research program, this is an impossible expenditure. For a government oceanographic agency with a constrained budget, it is difficult to justify against the competing demands of other research priorities. The Five Deeps expedition was funded by Vescovo's personal wealth. Most scientific programs do not have access to comparable resources. The comparison to space exploration that is often cited in arguments for increased ocean funding is imprecise but illustrative. NASA's annual budget is approximately $20 billion. The National Oceanic and Atmospheric Administration's entire budget, which covers weather forecasting, fisheries management, and ocean research among many other functions, is approximately $6 to $7 billion. The portion of that budget dedicated to deep-sea research is a small fraction of the total. The budgets of the few other nations with significant deep-sea research programs, Japan, China, France, Russia, are comparable to NOAA's ocean research allocation or smaller. The total global investment in HODL-specific research is difficult to quantify precisely, but is certainly measured in the tens of millions of dollars per year rather than the billions. Space science receives roughly 2 orders of magnitude more funding than HODL science. This is not an argument that space science is wrong or excessive. It is an observation that the allocation of scientific resources reflects something about human priorities and human psychology that is not simply calibrated to the scientific value of the questions being asked. The surface of the Moon and the surface of Mars are distant and romantic in a way that the bottom of the ocean, despite being equally exotic and equally unknown, apparently is not. The Hadal Zone does not look at us from the night sky. It does not catch the imagination in the same way. What we do not know about the Hadal Zone is, by any reasonable measure, more than what we do know. Less than 1% of the Hadal seafloor has been directly observed or sampled. The species richness of Hadal Trench communities is unknown because the sampling effort has been insufficient to generate species accumulation curves that plateau. Every new survey adds new species, suggesting that the total is much larger than the observed count. The biogeochemical processes operating in Hadal sediments, the rates of carbon mineralization, the nitrogen cycling pathways, the fate of the organic contaminants accumulating in Hadal organisms are known only in outline from a few intensively studied locations. Perhaps the least understood component of the Hadal ecosystem is the infaunal community, the organisms that live not on the sediment surface but within the top meter of sediment. In shallow marine sediments, the infauna includes an extraordinary diversity of organisms: nematode worms in densities of millions per square meter, polychaete worms of many species, small crustaceans, foraminifera, and bacteria in densities that dwarf those on the sediment surface. At Hadal depth, some infaunal organisms have been documented, but the sampling methods required to collect them, cores that penetrate the sediment surface and recover an undisturbed column of sediment for subsequent analysis, are difficult to deploy reliably at Hadal depth from a lander or an ROV. The sediment is soft, the currents near the bottom may push the core off target, and recovering the sediment column intact after a 12-plus-hour round trip through the water column requires engineering care that not all programs have invested in. What genomic surveys of Hadal sediment cores have revealed is that the diversity of microbial and meiofaunal life within the sediment may substantially exceed the diversity visible on the sediment surface. DNA sequences from Hadal sediment cores include many taxa not found in equivalent sediment cores from abyssal plains, suggesting that the Hadal sediment infaunal community has its own endemic species, as the megafaunal community does. But the physical organisms corresponding to many of these sequences have never been observed directly. The Hadal Zone will still be there. Long after the last expedition ship returns to port, after the cameras are serviced and stored, after the data has been processed and the papers written, the Hadal Zone persists unchanged. The pressure is constant. The darkness is constant. The temperature in the deepest parts of the major trenches hovers just above freezing, varying by less than a degree between the surface of the sediment and the water column 100 meters above it. Nothing in the Hadal Zone experiences a change of season, a change of light, a change of temperature corresponding to anything that happens at the surface. The world above could change fundamentally in its climate, its ocean chemistry, its biological composition, and the Hadal Zone would register those changes only slowly, as altered chemical compositions in the marine snow that takes months to reach the bottom, or as altered species compositions in the organic material that washes off continental shelves and eventually concentrates in the trench axes. The snailfish in the Izu Ogasawara Trench filmed once at 8,136 meters in 2023 by a camera on a wire, continues doing whatever snailfish do. It moves through water of 1,100 atmospheres pressure. It detects chemical gradients that indicate organic material somewhere in the trench. It moves toward those gradients, following information that travels through water in forms we have only begun to characterize. It feeds when it can and waits when it cannot. It is not aware of the camera that recorded it. It is not aware of the scientific literature that mentions it. It exists at a depth where no concept of observation applies. We know the snailfish is there because someone sent a camera down once. We know what it looked like for the few hours the camera recorded. We know its depth, approximately, and its species, approximately, and its approximate size. We do not know what it does for the 99.9% of its life during which no camera is present. We do not know how many other individuals share its section of the trench. We do not know what it eats when no bait has been lowered. We do not know how it reproduces or when or where its offspring go. This is the actual state of knowledge. It is not a failure of science. It is a precise measurement of how much exploration remains. The boundary between what is known and what is unknown is drawn at a very specific line, and that line lies just beyond the reach of the equipment that has been deployed so far. Move the line, build the instruments, fund the expeditions, make the dives, and the boundary retreats. It does not disappear. The deep ocean is large enough that the boundary will not disappear in any time frame we can currently imagine, but it retreats. There is something worth sitting with in that retreat. Every time a camera has reached a new depth, every time a lander has returned from a trench that had not been visited before, the record of what exists in those places has grown. Not shrunk, not simplified, grown and complexified. The Hadal Zone has not become less interesting as we have learned more about it. It has become more interesting because each answer has revealed 10 more questions, and each discovered species has implied the existence of others not yet seen. The ocean floor is not going anywhere. The trenches will continue to concentrate the falling material from above. The amphipods will continue to swarm whatever food arrives. The snailfish will continue to patrol the darkness at depths we can barely reach. And the questions we have about what all of this means, what the Hadal Zone is, fully in its biology and its chemistry and its geology and its connection to the rest of the ocean, will endure, waiting for the expeditions and the instruments and the time and the funding that will be needed to answer them. That is the nature of an unexplored place. The questions it holds are patient. They will wait as long as the darkness has waited, which is a very long time indeed. The story of how ocean depth came to be measured systematically is also the story of how human technology evolved to deal with a problem that has no easy analog on land. On land, you can walk to a mountain, measure its height with surveying instruments, and return with data. In the ocean, the floor is invisible from the surface, unreachable without specialized equipment, and in the deepest areas, unreachable by any means available until very recently in history. The earliest estimates of ocean depth came from sailors who dropped weighted lines over the side of their ships and measured how much rope they paid out before the weight touched bottom. In coastal waters and in relatively shallow seas, this method worked reasonably well. But in the open ocean, where depths were measured in miles rather than fathoms, the weight would often swing with current, and the line would curve, making accurate measurement impossible. The ropes themselves were long and heavy, requiring substantial crew effort to manage. A measurement in very deep water could take most of a working day and was subject to significant error. The Challenger expedition of 1872 to 1876 changed the quantitative ambition of ocean sounding without changing the fundamental method. The expedition was organized by the British Royal Society and funded by the Admiralty, and its scientific team was led by Charles Wyville Thomson, a Scottish naturalist who had argued for the expedition on the basis of his belief that the deep sea was not the biological wasteland that prevailing theory suggested it should be. He was right, and the expedition proved it, but the more lasting contribution was the systematic depth record it established. The Challenger's 492 deep soundings, however sparse on the scale of the ocean, constituted the first attempt to map ocean depth as a systematic scientific project rather than as a navigation aid. After the Challenger, the next major transformation in ocean measurement came in the early 20th century, driven by a practical need that had nothing to do with science. The sinking of the RMS Titanic in 1912 created intense interest in technologies that could detect icebergs at distance. Acoustic detection of underwater obstacles, using sound to locate things that could not be seen, was developed rapidly in the years after 1912, and the resulting technology, echo sounding, was applied to ocean depth measurement almost immediately. If a sound pulse could detect an iceberg at the same depth as a ship, it could equally detect the ocean bottom far below. The first successful echo sounder for deep water measurement was developed in the early 1920s. By the time the HMS Challenger 2, a second British oceanographic research vessel named after the original, made its 1950-1952 expedition, echo sounding had transformed the practical ability to measure ocean depth. The Challenger 2 expedition made thousands of soundings across the major ocean basins, including a measurement in the Mariana Trench area that was among the first accurate estimates of the depth of what would later be called the Challenger Deep, named in honor of both voyages. The expansion of echo sounding across naval and commercial shipping in the 1930s, 1940s, and 1950s produced, almost as a byproduct of navigation rather than science, an increasingly detailed picture of the ocean floor's topography wherever ships traveled. The mid-ocean ridge system was first clearly delineated during this period. Its existence inferred from the consistent pattern of elevated seafloor readings that appeared in the records of ships crossing the Atlantic. The geologist Marie Tharp, working at the Lamont Geological Observatory in New York, used echo sounder data from thousands of Atlantic crossings to produce the first comprehensive map of the North Atlantic seafloor in the early 1950s, revealing the rift valley running along the crest of the Mid-Atlantic Ridge and providing critical evidence for the theory of plate tectonics that was then still controversial. The multi-beam revolution of the 1970s and 1980s transformed ocean mapping from a linear track survey to an aerial survey, covering swaths of seafloor rather than single lines with each ship passage. A ship equipped with a multi-beam system crossing the Atlantic would now produce a strip of mapped seafloor rather than a single depth line. Crisscrossing the same area in multiple directions could produce a detailed bathymetric map of the entire region. The resolution of multi-beam mapping is limited by the depth and the physics of acoustic beam formation. In very deep water, the beam spread and resolution decreases. But even at its limits, multi-beam mapping produces data orders of magnitude more detailed than anything available from satellite-derived estimates. The satellite gravity approach that fills in the gaps between ship tracks is based on the observation that large seafloor features, mountains and trenches, create gravitational anomalies that can be detected from space as variations in sea surface height. The sea surface is not perfectly flat. It follows the geoid, the surface of equal gravitational potential, which bulges very slightly over massive underwater mountains and dips very slightly over deep trenches. These differences in sea surface height are small, on the order of millimeters to centimeters, but they can be measured by satellite altimeters with sufficient precision to infer the existence of major seafloor features. The resolution of this inference, however, is limited by the wavelength of the gravitational signal, which corresponds to features roughly 5 to 10 kilometers in horizontal extent. Anything smaller is invisible to this method. This resolution gap, the difference between what satellite gravity data can resolve and what multi-beam sonar can resolve, defines the fundamental information gap about the ocean floor. Satellite data tells us there is a mountain range here, a trench there, a basin in between. It does not tell us whether the mountain range has steep cliff faces or gentle slopes, whether the trench has a flat sediment plain at its bottom or a chaotic rockfall field, whether the basin is carpeted with smooth sediment or dotted with thousands of small volcanic cones. All of those details, the details at scales below 5 kilometers, are known only where ships have traveled with multi-beam equipment, which is a small fraction of the total ocean floor. The Hadal zone in particular suffers from this information gap because the trenches are geographically isolated and not on any shipping route. A ship traveling from Japan to Australia or from the United States to the Philippines does not cross the Tonga Trench or the Mariana Trench on any economical route. The surveys that have been done specifically target scientific research rather than being byproducts of commercial navigation. The total track length of multi-beam surveys within the Mariana Trench, for example, one of the most studied Hadal environments, is still small compared to the total length of the trench. Understanding this mapping history matters because it contextualizes every biological and geological finding from the Hadal zone. We find what we look for, and we can only look where we send instruments. The history of finding unexpected things in the deep ocean, hydrothermal vents, cold seeps, whale fall communities, the biological richness of seamounts, is partly a history of looking, and partly a history of finding things that were there all along but invisible to the instruments and the assumptions of an earlier era. The pressure that makes the Hadal zone so difficult to access is not a uniform barrier. It varies with depth, obviously, but it also varies in its effects depending on what material is experiencing it. The pressure at 10,000 meters, approximately 1,000 atmospheres, is not merely very high. It is high enough to change the physical behavior of many materials in ways that are not obvious from surface-level experience. Water itself compresses under high pressure. At 10,000 meters, seawater is roughly 4 to 5% denser than seawater at the surface, because the pressure has forced the water molecules slightly closer together. This compression has consequences for how sound travels through deep water. The speed of sound increases with pressure, and for how any air-breathing animal or any gas-filled structure would behave if brought to that depth. A balloon inflated at the surface would be compressed to a fraction of its original volume at 10,000 meters. A gas-filled swim bladder in a shallow waterfish would collapse completely. For engineering materials, the implications of 1,000 atmospheres are severe. Ordinary glass is strong in compression but brittle. It can withstand the uniform pressure of the deep ocean on external surfaces, but any imperfection, any internal stress concentration, any non-uniformity in the glass itself can initiate a crack that propagates catastrophically. The observation sphere of the Trieste used a plexiglass window not because plexiglass is stronger than glass, but because plexiglass deforms plastically before it fractures. It bends rather than shattering, which makes it safer in an application where a fracture would be immediately lethal. The design of the Trieste's window, tapered so that pressure forced the outer face more tightly into the hull, was a clever use of the environmental pressure as part of the sealing mechanism. Titanium's properties make it the dominant engineering material for deep-sea pressure vessels. Its yield strength, the stress level at which it begins to deform permanently, is significantly higher than that of most steel alloys at comparable weight. This means a titanium pressure vessel can be made with thinner walls than a steel vessel of equivalent strength, saving weight in an application where weight budget is critical. Its corrosion resistance in seawater is also superior to steel, which begins corroding rapidly in the oxygenated saline conditions of the upper ocean. At the Hadal zone's temperature and oxygen concentration, corrosion rates slow substantially, but any surface exposed to seawater for extended periods will eventually show degradation if not protected. Syntactic foam, the buoyancy material used in virtually all deep-sea research equipment, is a composite material consisting of hollow glass microspheres suspended in an epoxy matrix. The glass microspheres provide buoyancy because the air they contain is much less dense than water, and the glass walls are strong enough to prevent collapse under high pressure. The epoxy matrix holds the microspheres in a rigid structure that can be machined into any desired shape. But the compressive strength of syntactic foam is not unlimited. Standard syntactic foam formulations used in shallower research equipment will fail at Hadal pressures. The microspheres shatter and the material loses its buoyancy. Hadal-rated syntactic foam uses smaller, thicker-walled microspheres and higher-strength matrix materials to extend its pressure rating, but it is heavier and more expensive than standard foam. The interaction of these engineering requirements, high-strength titanium pressure vessels, Hadal-rated syntactic foam, pressure-compensated or oil-filled electronics, acoustic communication systems, power systems with sufficient energy density to operate for 12 to 24 hours at full depth, defines the cost and complexity of Hadal research vehicles. Each component must be individually tested at representative pressures before being integrated into a system. The integrated system must then be tested as a whole before its first operational deployment. The cost of pressure testing alone, using hyperbaric chambers rated for full ocean depth, is substantial. There are only a handful of such facilities in the world. The biological adaptations to this physical environment extend beyond the TMAO-based pressure adaptation of fish, and include a wide range of molecular, physiological, and behavioral strategies across the full diversity of Hadal life. The piezolite strategy used by fish is also used by marine invertebrates, but the specific compounds used differ between groups. Amphipods and other crustaceans use glycine betaine and other osmolytes in addition to TMAO. Different concentrations and combinations of piezolites characterize organisms from different depths and different taxonomic groups. At the cellular level, pressure adaptation requires changes not only in membrane lipid composition and protein folding stability, but also in the basic machinery of gene expression. The ribosome, the molecular machine that reads genetic instructions and synthesizes proteins, is sensitive to pressure. Surface-adapted ribosomes function less efficiently at high pressure. Hadal organisms have ribosomes with slightly altered structural and compositional properties that maintain function under the pressures of their habitat. These changes are subtle. A few amino acid substitutions in ribosomal proteins, adjustments in ribosomal RNA base pairing, but they are essential for the cell to maintain its normal rate of protein synthesis under pressure. The thermal environment of the Hadal zone compounds the pressure challenge. Bottom water temperature in the major Hadal trenches ranges from approximately 1 to 4 degrees Celsius, just above freezing, cold enough to significantly slow the biochemical reactions that drive metabolism. Cold temperatures favor pressure stability in some proteins. Cold-adapted proteins tend to have more flexible structures that resist the rigidity imposed by pressure, but they also reduce the rate of enzymatic reactions to the point where metabolism must be calibrated to function efficiently at very low temperatures. The combination of high pressure and low temperature represents a biochemical environment with no parallel in the surface biosphere, and organisms adapted to it have molecular machinery that is specifically tuned to this unique combination of conditions. The question of how organisms first colonized the Hadal zone is one that remains largely unresolved. The trenches are geological features that formed millions of years ago as their respective subduction zones became active. The organisms in those trenches therefore had millions of years to adapt to the Hadal environment, but the ancestors of those organisms came from shallower depths, from the abyssal plain or from the continental slope, and had to extend their depth range downward as each trench progressively deepened over geological time. The transition from abyssal to Hadal conditions involves a step change in pressure. Crossing the 6,000-meter threshold adds roughly 600 atmospheres to the pressure that would have required corresponding genetic changes in pressure-stabilizing biochemistry. For amphipods, the evolutionary history of Hadal colonization is partially readable in the genetic relationships among species at different depths. Molecular phylogenetics, the study of evolutionary relationships using DNA sequences, can in principle reconstruct the order in which different depth-adapted populations diverged from common ancestors. The data that exists suggests that Hadal amphipod lineages within any given trench system are closely related to Lycianassoid amphipod lineages from the surrounding abyssal plain, consistent with the idea that Hadal species descended from abyssal ancestors that gradually extended their depth range as the trench environment became available. The isolation of each trench system from the others has been maintained for long enough, typically tens of millions of years, that species from different trench systems are often phylogenetically distinct even when they occupy similar ecological roles. The Hirandelia gigas-dominated community of the Mariana Trench is not the same as the amphipod community of the Kermadec Trench, even though both are dominated by scavenging Lycianassoid amphipods at similar depths. The differences reflect both the phylogenetic distance between the founding populations of each trench and the subsequent evolution of each trench's community in isolation. This pattern is called allopatric speciation, the divergence of populations that have been physically separated from each other for long enough that genetic drift and local adaptation have made them reproductively incompatible. The question of dispersal, how Hadal organisms move between trench systems, if they do at all, is related to the question of colonization but distinct from it. If a Hadal trench were to be suddenly vacated by some catastrophic event, could organisms from another trench recolonize it? The answer depends on whether there is any pathway between trench systems at Hadal depth or whether organisms must cross shallower water to move between trenches. The abyssal plain that separates the Mariana Trench from the Tonga-Kermadec system reaches minimum depths of about 5,000 to 6,000 meters in the Central Pacific, right at or slightly above the Hadal threshold. Whether Hadal species can cross this passage, or whether they are biologically committed to depths below 6,000 meters and cannot survive even temporary excursion into the upper abyssal range, is not known. Some Hadal species appear to have wider depth ranges than others. Hirandelia gigas has been collected from depths ranging from roughly 4,000 meters to the full depth of the Mariana Trench, suggesting it can tolerate a substantial pressure range. Other species appear to be confined to very narrow depth ranges within trenches, suggesting tight pressure optimization. The range of depth tolerance among Hadal species has implications for both their dispersal potential and their vulnerability to the kind of disturbance that comes from earthquake-triggered sediment events or from future changes in bottom water chemistry driven by climate change. The relationship between the Hadal zone and climate change is an area of active research and significant uncertainty. The deep ocean absorbs heat and carbon dioxide from the atmosphere through surface processes, and those inputs are eventually transported to the deep ocean through thermohaline circulation. The global pattern of deep water formation and movement driven by density differences between water masses of different temperature and salinity. Changes in ocean warming, freshening, and stratification at the surface can alter the rate of deep water formation, potentially affecting the chemistry of the deep ocean on timescales of decades to centuries. For the Hadal Zone specifically, potential climate impacts include changes in the oxygen content of deep water, changes in the temperature of Hadal bottom water, and changes in the flux and composition of organic material arriving at the trench floor as surface productivity patterns shift. Whether any of these changes are large enough to significantly affect Hadal biological communities on timescales shorter than geological is not known. The Hadal Zone has been a stable environment on timescales of millions of years, and its biology has been shaped by that stability. Relatively rapid environmental changes, rapid in geological terms, meaning centuries to millennia rather than millions of years, represent a regime that Hadal communities have limited experience with. The monitoring infrastructure needed to detect environmental change in the Hadal Zone does not exist at present. There are no permanent observatories in any Hadal trench. No moored instruments record continuous data on temperature, chemistry, current, or biological activity from any Hadal site. The data that exists from the Hadal Zone comes from episodic expeditions, discrete visits separated by months or years, and from the landers that individual research programs deploy for hours or days before recovering them. Building a continuous monitoring capability at Hadal depth would require technological developments that have not yet been made and funding commitments that have not been secured. The contrast with other extreme environments is instructive. The summit of Mount Everest is monitored continuously by weather stations. The surface of Mars is monitored continuously by orbital satellites. The deep Antarctic ice sheet is monitored by automated instruments deployed during expensive expeditions and left to record data for months or years before being recovered. All of these environments are less accessible than the Hadal Zone in some respects and more accessible in others. The deep Antarctic ice is cold and remote, but requires no pressure-rated instrumentation. The surface of Mars requires no instruments at all to be brought back. Data can be transmitted by radio. The Hadal Zone combines physical inaccessibility, extreme pressure requirements, and the impossibility of radio communication in a combination that makes continuous monitoring technically challenging in ways that these other environments are not. What exists instead of continuous monitoring is an accumulating but still sparse record of episodic observations, each representing a point in time and a point in space in a vast and varied environment. The picture assembled from these observations is real and valuable, but it is necessarily an approximation of the actual biological and chemical state of the Hadal Zone at any given moment. The organisms visible to a camera during a 6-hour lander deployment are the organisms that happen to come near that location during that interval. The chemistry measured by a water sampler lowered on a wire reflects the conditions at that depth on that day. The sediment collected by a corer represents the accumulated material at one specific spot on a trench floor that may cover thousands of square kilometers. Assembling a comprehensive picture of the Hadal Zone from these sparse observations requires statistical inference, reasoning from what has been seen to what is likely to be present more broadly, and that inference is only as reliable as the sampling design that produced the observations. If landers are deployed preferentially at points in a trench where the floor is flattest and most accessible, the picture that emerges will be biased toward the biology of flat trench floors and will underrepresent the biology of the steep walls, the rocky outcrops, and the areas where sediment disturbance is highest. Designing sampling programs that avoid this kind of systematic bias at Hadal depth is difficult because the physical access limitations mean that researchers cannot simply choose the most representative sampling locations with the flexibility they would have in shallower water. The role of the Hadal Zone in global biogeochemical cycles is another area of substantial uncertainty. The ocean is the largest reservoir of dissolved inorganic carbon on Earth's surface. It contains roughly 50 times as much dissolved carbon dioxide as the atmosphere. The deep ocean, including the Hadal Zone, is a significant long-term sink for this carbon. Organic material sinking from the surface is partly mineralized back to carbon dioxide by bacteria as it sinks, but a fraction of it reaches the deep sea floor and is incorporated into the sediment, where it is effectively removed from the active carbon cycle on timescales of thousands to millions of years. The Hadal Zone, because it concentrates organic material from a larger catchment area than its size would suggest, may contribute disproportionately to this long-term carbon burial. But quantifying this contribution requires knowing the rates of organic carbon deposition, burial, and mineralization at Hadal depths, which are known only approximately from a handful of sites. Nitrogen cycling in the Hadal zone is similarly undercharacterized. The fixed nitrogen that organisms need to build proteins and nucleic acids is cycled through multiple forms: ammonium, nitrate, nitrite, dissolved organic nitrogen, by bacteria and archaea that perform nitrification, denitrification, and anaerobic ammonia oxidation. In the oxygen minimum zones that occur in the mid-water column of some ocean basins, these nitrogen cycling processes are particularly active and have large effects on the availability of fixed nitrogen to surface organisms through upwelling. At Hadal depth, the oxygen concentrations are low but not anoxic in most trenches, and the nitrogen cycling community includes both aerobic and anaerobic metabolizers. The relative rates of these processes at Hadal depth and their net effect on nitrogen cycling in the broader ocean system have barely been measured. The question of what the Hadal zone is contributing to the ocean's chemistry, rather than simply what lives there, is in some ways the most significant scientific gap, because it connects the Hadal zone to processes that affect the entire ocean and ultimately the atmosphere and climate. A complete understanding of the ocean carbon cycle, which is fundamental to understanding climate change and to predicting how the ocean will respond to human emissions of carbon dioxide, requires knowing what happens to organic carbon once it reaches Hadal depth. That knowledge is still in its early stages. All of these gaps in knowledge are not permanent features of the situation. They are limitations of the present moment, determined by the equipment that exists, the funding that has been secured, and the human attention that has been directed toward Hadal science. Each of these factors is changeable. Technology for deep ocean operations continues to improve. The cost of autonomous vehicles has been declining as the technology has matured and as more manufacturers have entered the market. Battery energy density has been improving steadily, extending the endurance of autonomous systems. Pressure-rated electronics have become more capable and less expensive as the materials and manufacturing techniques have been refined. None of these improvements are dramatic on a year-by-year basis, but they compound over decades into substantial changes in what is technically achievable. The future of Hadal science will likely be shaped more by autonomous systems than by crewed vehicles. The economics are clear. A crewed vehicle requires a complex life support system, careful management of descent and ascent profiles to avoid decompression injuries, and the mental and physical constraints of human operators working in an extremely confined space under extreme conditions. An autonomous vehicle, or a fleet of autonomous vehicles, can stay at depth for as long as its batteries last, can work at full depth continuously, and can be programmed to survey larger areas with more systematic coverage than any single crewed dive can achieve. The scientific capabilities foregone by removing the human from the vehicle, the real-time decision-making, the experienced eye that notices an unexpected detail, the flexibility to deviate from a pre-programmed survey when something interesting appears, are significant. But they are being progressively recovered by improvements in machine learning and computer vision that allow autonomous vehicles to make increasingly sophisticated decisions without human input. A fleet of small, inexpensive autonomous landers capable of reaching Hadal depth, each covering a different location in a trench simultaneously, could produce a spatial picture of Hadal biological communities that no single vehicle program can approach. A persistent autonomous vehicle capable of operating at Hadal depth for days or weeks, surveying systematically and returning to a base station to recharge and upload data, could provide a temporal picture of biological activity that no episodic expedition can provide. Neither of these technologies fully exists today, but the components exist, and their assembly into operational systems is a question of funding and engineering effort rather than fundamental scientific impossibility. The snailfish, the amphipods, the piezophilic bacteria, the DSAG archaea, the holothurians sifting through sediment contaminated with PCBs from industrial processes that ended decades ago on the surface of the Earth. All of these are present in the Hadal zone right now, doing what they have always done in a darkness and pressure that humans have reached only a handful of times in history. The scientific record of their existence is the record of those handful of visits. It is partial and incomplete in proportion to the rarity of the visits. More visits will produce a more complete record. The questions that will be answered by more visits are the questions that determine what the Hadal zone actually is, in its full biological, chemical, and geological complexity, rather than in the sketched outline that is all the current record provides. The darkness at 11,000 meters is the same darkness that has been there since the trench formed. The pressure is the same pressure. The temperature is within a degree of what it has been for the past several million years. What changes is our knowledge of it, and that knowledge is built one expedition at a time, one lander deployment at a time, one DNA sequence at a time, accreting slowly like the sediment that falls through the water column, and eventually, after weeks of descent, comes to rest on the trench floor and becomes part of the landscape that the next camera will illuminate. That camera will find something new. It always does. The Hadal Zone has never been visited without yielding a discovery that was not anticipated before the visit. The record of unexpected things found at depth is long and consistent enough to constitute a principle. There is more in the deep ocean than the models predict, and each visit revises the models upward. There is no reason to expect this pattern to end. The discovery of unexpected things in the Hadal Zone is not a feature of early exploration that will cease as the zone becomes better known. It is a feature of an environment so incompletely sampled that almost any measurement constitutes new information. That situation will not change until the sampling becomes comprehensive. Comprehensive sampling of the Hadal Zone is a project that has not yet seriously begun. This is the state of things. Accurate as of now. The trenches are there. The organisms are there. The processes are happening. The questions are open, and the work of answering them waits for the expeditions that have not yet been funded, the vehicles that have not yet been built, and the scientists who have not yet turned their attention to the deepest places on Earth, drawn down by the same irresistible logic that drives all exploration. The simple, stubborn fact that there is something there, and we do not yet know what it is. The transition zone between the abyssal plain and the Hadal Zone, the depth range from roughly 4,000 to 6,000 meters, is itself an understudied environment that bridges two different biological worlds. Organisms in this transitional zone experience pressures intermediate between the abyssal and the Hadal, temperatures that are cold but not at the extreme cold of the deepest trenches, and food availability that reflects the abyssal plain's dependency on marine snow without the geometric concentration effect of the trench axis. The biological communities in this transition zone are not simply intermediate versions of abyssal or Hadal communities. They are distinct assemblages with their own characteristic species, their own patterns of abundance and diversity, and their own ecological dynamics. The transition zone is important for understanding how organisms move between the abyssal plain and the Hadal zone. If Hadal species evolved from abyssal ancestors, as the phylogenetic evidence suggests, they must have passed through this transitional depth range at some point in their evolutionary history. Whether they passed through gradually, over millions of years of incremental depth range extension, or through episodic colonization events following tectonic opening of new trench environments, is a question that cannot be answered from the current data. But the organisms living in the transition zone today may represent lineages that are currently in the process of extending their depth range, or lineages that once occupied the Hadal zone and have retreated, or lineages that have been stable at their current depth for geological time. The abyssal plain at its deeper margins, the areas where the plain transitions into trench walls at depths around 5,000 to 6,000 meters, has rarely been the specific focus of biological surveys. Most abyssal surveys target depths between 3,000 and 4,500 meters, where the plain is flattest, most accessible, and most representative of the dominant abyssal habitat type. The deep margins of the plain, where it begins to slope into the trench, are geometrically complex, harder to sample safely, and have therefore been systematically avoided in most programs. The organisms that live there are largely unknown. The biological communities of the Hadal trenches are not simply organized vertically, by depth. They are also organized horizontally, within each trench, in ways that reflect the physical structure of the trench itself. A Hadal trench is not a uniform environment. The floor of the trench, the axis, the deepest part, is flanked by walls that rise steeply at angles of 20 to 40 degrees before merging with the surrounding abyssal plain. The walls are composed of rock, either oceanic basalt from the subducting plate or sedimentary material from the continental slope above, and their biological communities differ from those of the flat axis below them. The transition from steep rocky wall to soft sediment floor is itself a habitat boundary, with different organisms on either side. On the rocky walls of Hadal trenches, organisms that need a hard substrate for attachment, cold water corals, sponges, crinoids, serpulid worms, and calcareous tubes, can be found at depths that exceed anything in the abyssal plain, because the trench walls provide what the flat sediment floor does not. These communities have rarely been photographed in detail, because most lander deployments target the flat trench axis, where landing is predictable and safe, rather than the steep walls, where a lander might slide or tumble. ROV surveys of trench walls would require vehicles capable of holding position in mid-water against the deep currents that move along trench flanks, and would need to cover very large vertical extents to document the full depth range of the wall community. The trench axis itself shows spatial variability that reflects the history of sediment input. Areas where turbidite deposition, the mass transport of sediment from shallower areas into the trench during submarine landslides, has been recent, are dominated by fresh, organic-rich sediment that has not yet been heavily colonized. Areas where the last major disturbance was further in the past show more developed biological communities, with higher organism densities and greater diversity. Areas permanently exposed to strong bottom currents, where fine sediment cannot accumulate, may be completely bare of any biological activity visible at camera scale. The spatial pattern of the Hadal floor community is a mosaic, reflecting the geological history of each location rather than a uniform biological zone. The Japan Trench provides the best documented example of how geological events reshape this mosaic. Before the 2011 Tohoku earthquake, the Japan Trench had been surveyed by JAMSTEC's Shinkai 6500 and by various lander programs, producing a baseline picture of the biological community at several depths. When the earthquake struck, the seafloor along the Japan Trench was subjected to violent shaking. Slopes that had been stable for decades or centuries failed and collapsed, sending plumes of sediment billowing down into the trench axis. Material that had been slowly accumulating on the lower slopes for years was mobilized in seconds and deposited in the trench within hours. The post-earthquake surveys by JAMSTEC found a dramatically altered landscape. The areas of greatest disturbance showed bare, freshly deposited sediment with no large organisms visible. The existing community had been buried and killed by the rapid deposition. In the months and years following the earthquake, those areas began to show signs of recolonization. First bacterial mats on the fresh sediment surface, then small polychaete worms, then amphipods exploiting the organic material in the fresh deposit. The sequence of recolonization observed in the Japan Trench is the closest thing to an experimental test of Hadal community dynamics that researchers have had, a natural experiment in which a community was effectively reset to zero and the subsequent recovery tracked through time. This recovery process is informative about the resilience of Hadal communities. The organisms that recolonize disturbed areas must come from somewhere, from undisturbed areas of the same trench, from the adjacent abyssal plain, or from the water column if their larvae can survive extended periods at depth. The speed of recolonization depends on how far the nearest undisturbed populations are, how mobile the colonizing species are, and how favorable the disturbed area is as a habitat once the initial sediment has settled. In the Japan Trench, full biological recovery to something resembling the pre-earthquake community is expected to take decades, based on what is known about the growth rates and colonization rates of the organisms involved. The Philippine Trench, which runs along the eastern coast of the Philippine Islands and reaches depths of approximately 10,540 meters at its deepest point, the Galathea Deep, has received less systematic study than the Mariana, Tonga, or Kermadec trenches. It is in one of the most seismically active parts of the Pacific Ring of Fire, located at the junction of the Philippine, Eurasian, and Pacific plates, where the geometry of plate interaction is particularly complex. The biological surveys that have been done suggest a community broadly similar to other Western Pacific Hadal systems, with snailfish and lysianosoid amphipods dominating the megafaunal observations, but the database is too sparse to characterize the community in anything like the detail available for the Mariana system. The Kuril-Kamchatka Trench, which extends from the Kamchatka Peninsula in Russia southward, along the Kuril Island arc toward Japan, reaches approximately 10,500 meters at its deepest point. Soviet oceanographers made early deep-sea research contributions in this trench during the Cold War, when Soviet oceanographic programs were active and well-funded. Some of the earliest biological samples from Hadal depths in this trench date to the 1950s and 1960s, collected by Soviet research vessels. The species described from those early collections remain among the known hadal fauna of the trench, but subsequent systematic surveys have been limited, and the current state of the biological community is not well characterized. The deepest point of the Indian Ocean lies in the Java Trench, also called the Sunda Trench, at approximately 7,187 meters at the Sunda Deep. This is substantially shallower than the Pacific Hadal systems, placing the deepest Indian Ocean environment at the margin of the Hadal zone rather than at its extreme. Victor Vescovo's dive to the Java Trench in April 2019 as part of the Five Deeps expedition represented the first crewed descent to the deepest point of the Indian Ocean. The footage from that dive showed a sediment-covered floor with amphipods visible near the lander bait and a generally similar appearance to other trench floors. Though the absence of a dedicated biological survey program in this area means that the community there is described only from the limited footage collected during the dive. The Southern Ocean Hadal environment is represented primarily by the South Sandwich Trench, which runs along the boundary between the South American and Scotia plates northeast of the South Sandwich Islands, reaching depths of approximately 8,266 meters at the Meteor Deep. Victor Vescovo's dive to the South Sandwich Trench in February 2019 was conducted in extremely challenging surface conditions. The Southern Ocean in the austral summer is one of the most consistently rough sea environments on Earth, and the biological observations from the dive showed organisms typical of sub-Antarctic deep-sea environments, including some species not previously recorded from that depth. The Arctic Ocean's contribution to Hadal science is the Malloy Deep, located in the Fram Strait between Greenland and Svalbard at approximately 5,551 meters. At this depth, the Malloy Deep is below the Hadal threshold of 6,000 meters only by some definitions. Some researchers use 6,000 meters as the boundary, others use different criteria. Vescovo's dive to the Malloy Deep in August 2019 was the shallowest of the five deeps in absolute terms, reflecting the relative shallowness of the Arctic Ocean compared to other ocean basins. The biological community of the Malloy Deep is influenced by the cold, dense water masses that form in the Arctic and sink into the deep basins. The most oxygen-rich deep water in the world ocean forms in the Arctic, and is distinct from both Atlantic and Pacific Hadal communities in ways that reflect this different water mass history. The scientific programs that have studied the Hadal zone have been conducted by a small number of institutions in a small number of countries. JAMSTEC in Japan, with its Shinkai 6500 submersible and its extensive lander programs, has produced much of the photographic and biological record of Pacific Hadal communities. The University of Hawaii's Hades project contributed systematically to the comparative biology of the Mariana and Kermadec trenches. Newcastle University's Deep Sea Lander program, under Alan Jamieson and his colleagues, produced the contamination data and much of the Scottish biodiversity data. The University of Western Australia has operated lander programs targeting fish community depth limits. The Woods Hole Oceanographic Institution in the United States has contributed to Hadal research through its development of the Nereus hybrid ROV and its participation in various expedition programs. These institutions represent a tiny fraction of the world's ocean research capacity. Most of the countries that border the Pacific Ocean, with its concentration of deep trenches, have limited or no Hadal research programs. The Philippines, Indonesia, Tonga, Fiji, Papua New Guinea, and most of the Pacific Island nations have deep trenches within or adjacent to their exclusive economic zones, but essentially no capability to study them. The biological and chemical inventory of the Hadal zone is therefore not only sparse in absolute terms, but also geographically biased toward the trenches nearest to the institutions that have had the resources and technology to study them. This geographical bias has scientific consequences. The trenches near Japan have been studied more than the trenches near South America. The Mariana Trench has been studied more than the Philippine Trench, despite their similar depths and proximity. The Kermadec Trench has received more attention than the Tonga Trench, despite the Tonga being deeper, partly because New Zealand's oceanographic programs have focused on the Kermadec as the nearest major Hadal system. The result is that our understanding of Hadal biology is shaped not only by what lives in the Hadal zone, but by which parts of the Hadal zone happen to be nearest to research institutions with the resources to go there. Correcting this bias requires not only more resources, directed toward Hadal research generally, but also a deliberate effort to extend surveys to the less studied trench systems. Proposals for systematic surveys of understudied Hadal trenches have been made in the literature and in funding applications, but the resources required for such surveys, ships with the appropriate deep-sea capability, lander systems rated for full Hadal depth, Experienced scientific teams are not readily available in the countries where the unstudied trenches are located. The internationalization of Hadal science, in which institutions from well-resourced countries partner with institutions from the countries adjacent to understudied trenches, has the potential to address this gap. But building such partnerships takes time and requires sustained commitment from multiple parties. The deep ocean in general, not just the Hadal zone, faces a fundamental funding asymmetry that reflects something about how science and government prioritize research investment. The deep sea offers no immediate economic return. It does not contain resources that can be extracted at current prices with current technology, with the partial exception of the polymetallic nodules and seafloor massive sulfide deposits that deep-sea mining companies have been evaluating for decades without yet bringing a mine to commercial operation. It does not offer tourism or recreation. The scientific value of understanding the deep ocean, its role in climate regulation, its biological diversity, its pharmaceutical potential, its contribution to biogeochemical cycles, is real, but distributed and long-term rather than concentrated and immediate. The pharmaceutical potential of deep-sea organisms, including Hadal species, is worth specific mention because it represents one of the more concrete cases for the instrumental value of deep-sea biodiversity. The deep ocean has already produced several compounds of pharmaceutical significance. Halochandrin B, isolated from the deepwater sponge Halochandria ocadii, led to the development of eribulin, an anti-cancer drug approved by the U.S. Food and Drug Administration in 2010 for the treatment of metastatic breast cancer. Discodermalide, isolated from the sponge Discodermia dissoluta collected from the Caribbean seafloor at depths between 30 and 200 meters, has anti-tumor activity and has been studied as a potential treatment for drug-resistant cancers. These examples come from organisms in relatively shallow deep-sea environments compared to the Hadal zone. Whether Hadal organisms, with their biochemistry tuned to extreme pressure and temperature, contain compounds of comparable pharmaceutical significance is unknown, because Hadal organisms have rarely been collected in quantity sufficient for chemical screening. The challenge of obtaining Hadal organisms for pharmaceutical screening illustrates the broader challenge of Hadal biological research. Even getting organisms to the surface in a condition suitable for biological and chemical analysis is difficult. Organisms collected in traps are often dead by the time they surface, having been crushed as the trap is raised through decreasing pressure during the ascent, or having been killed by the temperature change as the cold Hadal water gives way to warmer mid-water temperatures. Organisms collected in jars or preservative solutions are chemically altered by the fixative. The development of pressurized recovery systems, containers that maintain Hadal pressure during ascent and can be opened in a high-pressure laboratory on the ship, is technically possible, but adds enormously to the complexity and cost of a sampling operation. The instruments and methods needed to fully characterize the Hadal zone do not all exist yet in their final form. Some of them, better autonomous vehicles, improved environmental DNA methods, pressurized recovery systems, are in development. Others, continuous monitoring systems capable of operating at Hadal depth for months or years, genomic databases comprehensive enough to identify all Hadal organisms from environmental sequences, models of Hadal biogeochemistry validated against comprehensive measurements, require scientific progress that will take years or decades to achieve. The Hadal zone is not simply an exploration problem. It is a measurement problem and a modeling problem and a biological problem and a chemical problem, all intertwined and all requiring simultaneous progress. The progress that has been made, when viewed against the scale of what remains unknown, can seem modest. When viewed against where the field was 30 years ago, when Hadal biology consisted of a handful of species described from samples collected by early Soviet and Danish expeditions, a few photographs taken by the Kaiko ROV, and no understanding whatsoever of the chemical and microbial ecology of these environments, the progress seems substantial. The field of Hadal science has grown from a curiosity to a legitimate discipline with its own journals, its own methodological standards, its own community of researchers, and its own set of open questions defined precisely enough to be answerable in principle. The open questions that define Hadal science today are different in kind from the open questions that defined it a generation ago. A generation ago, the fundamental questions were, what lives down there? Are the trenches as biologically barren as the abyssal plains? Is the Hadal zone simply more of what is found above, or is it qualitatively different? Those questions have been answered broadly. The Hadal zone contains a distinctive fauna. It is not simply deep abyssal. The trenches are not barren. The next generation of questions is more specific and more quantitative. How many species are there specifically? What are the rates of organic carbon processing? How does the Hadal Zone function as part of the broader ocean system? What are the mechanisms of pressure adaptation at the molecular level for specific proteins and specific organisms? These questions require the next generation of methods to answer. The patience required to answer them is the patience of the deep ocean itself. The Hadal Zone accumulated its sediment record over millions of years, one marine snow particle at a time, one turbidite deposit from one earthquake at a time, one whale fall in each decade of each million years. The scientific record of the Hadal Zone is accumulating by similar increments, one lander deployment at a time, one expedition at a time, one DNA sequence at a time. The database grows. The picture sharpens. The questions become more specific as the earlier, broader ones are resolved. The process continues at its own pace, constrained by funding and technology, and the difficulty of the environment, advancing by the persistent effort of researchers who find in the Hadal Zone exactly what all deep environments have offered, a place where ordinary assumptions do not hold, and where the reward for extraordinary effort is, without exception, surprise. The ocean floor is patient. The trenches hold their inventory quietly, as they have always done, as they will continue to do long after any camera has been there and gone. What we have seen of the Hadal Zone is real and valuable. What we have not seen is larger by every measure. The work of seeing it, of extending the record, of sharpening the picture, of asking and answering the specific questions that the current picture makes possible, is the work of the coming decades. It is work worth doing. The place is there, and it is waiting, not with the patience of something aware, but with the patience of depth and darkness and pressure and time, which is the most complete patience there is. And now, as the distance between the surface and the deepest trenches settles into your awareness as something real rather than abstract, 11 kilometers of water, 11 kilometers of pressure and darkness and life. Let the weight of all that depth carry you down into your own quiet place. The ocean will still be there when you wake. The snailfish will still be moving through its world of pressure. The amphipods will still be waiting. None of it requires your attention to continue. Let it go on without you for now and rest.